please welcome the group from Fluvanna County High School.
Hi, my name is Newland Humphrey alongside Libby Hartung, Cameron Odell, and Francis Fulton, and we are from Fluvanna, and we are testing the effects of ethanol on wild-type and mutant Arabidopsis thaliana that lack the AADL gene.
Arabidopsis thaliana is a small flowering plant that is part of the mustard family. It was originally native to Eurasia, but as it's a common weed now, it is found in Africa and the United States as well. Prime conditions for Arabidopsis to grow in is partially shaded areas, but it can survive in a variety of conditions such as sandy soil, clay soil, and it is also easily maintained in a lab environment. Arabidopsis serves as a model for genetic research because of its small genome size and is able to be fully sequenced. The first time Arabidopsis was sequenced completely was in 2000 by the Arabidopsis Genome Initiative. Arabidopsis has a short lifespan of approximately 6 to 8 weeks, and it can be genetically altered with relative ease when using Agrobacterium to infections. The purpose of the following experiment is to test the effects of ethanol on the AADL gene.
Now, the independent variable that our class chose was ethanol. Ethanol is produced and consumed by countless organisms. Ethanolic fermentation is thought to be an explanation for energy production in anaerobic conditions, such as the early atmosphere. Excessive ethanolic fermentation is thought to trigger stress responses in plants and also trigger flavonoid biosynthesis. The varying concentrations of ethanol that we tested are on our Arabidopsis plants act as a mode of stress that will affect flavonoid biosynthesis in the Arabidopsis. The class hypothesized that as the concentrations of ethanol increase from 1% to 5% to 10% in the water given to the Arabidopsis, that the pigment of the mutated plant that lacks the AADL gene will become darker than in the wild type.
We used the following materials and methods to make sure our experimental data was valid. We planted the Arabidopsis seeds using a pipette in small containers containing soil and fertilizer. These plants were then watered and then placed under a humidity dome. Later, the dome was cracked and then removed once the seeds had fully sprouted. On that day, we also thinned the plants, so there were only 5 plants per spot. There were 8 plots in total, with 4 containing wild-type Arabidopsis and 4 containing mutant. In each set, there was 1 control, and then 3 different concentrations of ethanol, 1%, 5%, and 10%. 3 days after this, the plants were watered again and measured, and then 4 days after, were given the first treatment of ethanol. This was repeated every 5 days, with us working around the weekends when we had to. The plants were measured for the rosette width, bulb height, and how dark the leaves were.
During our experiment, the first week we only measured and watered the plants, and then during the second week we started to treat our plants with ethanol, and the control group was not affected and had the most growth and the least stress because they were not supposed to be effective of ethanol. The 1% had very little stress and had very similar measurements to the rosette width of the control group. And the 5% and the 10% were the most affected by the ethanol and were very stressed out. And towards the end of the experiment, they started to die because the ethanol chemicals. And this is, the wild type had larger rosette widths because the AALD gene was not removed from the plant. And this gene helps the plants better undergo stress and grow more because the AALD gene helps the ribose to create protein.
The darkness and and purpleness of the leaves was the main topic in our experiment. As you can see on this graph, green being mutant Arabidopsis and purple or gray, I can't tell, being wild type Arabidopsis. As you can see here, even in the control, the mutant type Arabidopsis continued to go darker and darker over time, while the control remained the same. A similar trend is shown on the 1% graph, with the mutant type Arabidopsis being more darker than the wild type. As you can also see, the graphs go down within the last few weeks of the measurements because the plants begin to die from all the stress that ethanol was put on. Again, the same trend is shown in 5% and also the 10%. This trend remained true throughout our experiment except for one measurement in 10%.
As the data presented has shown, our hypothesis was concurrent with our experimental results. The AADL gene is thought to reduce stress the AADL gene is thought to aid in reducing stress caused by ethanol.
So to reduce the experiment, we concluded that the Arabidopsis should have been placed under 10 hours of light rather than 23 hours of light, because 10 hours of light is a more natural light cycle that would reduce stress in the Arabidopsis. Both groups of control plants ended up acquiring a small amount of purple pigment due to the unnatural cycle of light. Several plants also went missing due to mistakes made by plant maintenance. And 3 groups of plants also contracted mold due to contaminated rulers that were used when measuring the rosette width and mold height. And for better qualitative, more precise qualitative data results, different shades of purple should have been gathered and then had the qualitative data based off of those images, because each student perceived the amount of purple pigment in the leaves differently, therefore the results were not as accurate. The research that has been done could have future applications in the medical fields and in agricultural food production. Increased flavonoid intake has been linked directly with increased bone density in a study done by the Linus Pauling Medical Institute. Flavonoids also are very high in antioxidant compounds, which can lead to disease prevention in chronic diseases such as cardiovascular disease, neurodegenerative disease, and cancer. And for cancer treatments, flavonoids are most likely predicted to be beneficial in the cases of ovarian, breast, pancreatic, and colon cancer. They could play a big role in advancements in chemotherapy. And flavonoids can also assist in creations of new anti-inflammatory medications and immune system benefits. And in the American agricultural industry, flavonoids act as a natural insecticide, which could be a big benefit for many organic farmers in America.
So you guys go back, so it doesn't matter which graph any of them.
Now, I do want to specify that these were data. This was the data taken from one group, just because due to the outside variables that I explained earlier, the overall average was not very concise. They didn't really agree with each other. So in order for all of you to get a better look at what really happened in the experiment, we just took the data from one group.
So what I wanted to see, and you guys can maybe say it better than I can see it, is sometimes what I look for is, are the mutant plants or the experimental plants or anything, are they just lagging behind the wild type counterparts, or is the mutant just lagging behind the wild type and it eventually gets to the same point, or is it just staying in a different pattern the entire time? And so what I wanted to see, sometimes I just can't do it for some reason, did you see the pigmentation ended up being the same eventually? It just took longer to get to that point? Or was it always just different consistently the whole time? Always a lighter color the whole time, never made it to that same point?
Consistently, the mutant was darker than the wild type, but towards the end, what happened was as the plants started to die, the pigment, of course, it was lacking since the leaves were losing their chlorophyll. And that was taken into account, but for the most part, they were different the entire time. And towards the end, they kind of caught up to each other. But for the most part, the mutant stayed darker the entire time throughout the entire experiment.
How did you convert the qualitative data into quantitative, like numerical?
Oh, so our numerical data was bolt height and rosette width, and then our qualitative data was the amount of purple pigment in the leaf.
Yeah, but like, how did you
I
convert the?
I apologize. So what happened was we measured the qualitative data on a scale from 0 to 10, 0 being the leaves are very green and lush, and 10 being they're very dark and purple pigment.
Can we go back to the control? So